Good morning, viewer. Welcome to our Glory Globe TV. Yes, here's the breaking news. Huge libraries of drug compounds may hold potential treatments for a variety of diseases such as cancer or heart disease. Ideally, scientists would like to experimentally test each of these compounds against all possible targets, but doing that kind of screen is prohibitively time consuming. In recent years, researchers have begun using computational methods to scan those libraries in hope of speeding up drug discovery. However, many of those methods also take a long time, as most of them calculate each target protein's three-dimensional structure from its amino acid sequence, then use those structures to predict which drugs molecules it will interact with. Researchers at MIT and Tufts University have now devised an alternative computational approach based on a type of artificial intelligence algorithm known as a large language model. This model's one well-known example is ChatGPT can analyze huge amounts of text and figures out which words, that is called in the case amino acid, are most likely to appear together. The new model known as PLEX can match target proteins with potential drug molecules without having to perform the computationally intensive step of calculating the molecule's structures. Using these methods, the researchers can screen more than 100 million compounds in a single day, much more than any existing model. This work addresses the need for efficient and accurate in silico screening of potential drug candidates, and the scalability of the model enables large-scale screens for analyzing of target effects, drug repurposing, and determining the impact of mutations on drug binding, says Bonnie Berger, the Simons Professor of Mathematics, head of the Computation and Biology Group in MIT's Computer Science and Artificial Intelligence Laboratory, and one of the senior authors of the new study. Lenore Crown, a professor of computer science of Tufts University, is also a senior author of the paper which appears this week in the Proceedings of the National Academy of Science. Rohit Singh, a CSAIL research scientist, and Samuel Seljeski, an MIT graduate student, are the head authors of the paper, and Byron Bryson, an associate professor of biological engineering at MIT and a member of the Ragon Institute of MGH, MIT and Harvard, is also an author. In addition to the paper, the researchers have made their model available online for other scientists to use. In recent years, computational scientists have made great advances in developing models that can predict the structures of proteins based on their amino acid sequences. However, using these models to predict how a large library of potential drugs might interact with a cancerous protein, for example, has proven challenging mainly because calculating the three-dimensional structures of the proteins requires a great deal of time and computing power. An additional obstacle is that these kinds of models don't have a good track record for eliminating compounds known as decoys, which are very similar to a successful drug but don't actually interact well with the target. One of the long-standing challenges in the field has been that these methods are fragile, in the sense that if I gave the model a drug or a small molecule that looked almost like the true thing, but it was slightly different in some subtle way, the model might still predict that they will interact even though it should not says. Researchers have designed models that can overcome this kind of fragility, but they are usually tailored to just one class of drugs molecules, and they are not well suited to large scale screens because the computation takes too long. The MIT team decided to take an alternative approach based on a protein model they first developed in 2019, working with a database of more than 20,000 proteins. The language model encodes this information into meaningful numerical representation of each amino acid sequence that capture association between sequence and structure. Beyond this report, Globe TV, Delhi, India.